Imagine a world with no oxygen, no animals, no plants, just ancient oceans, boiling rocks, and the first fragile whispers of life. Now imagine reaching back across 3 billion years and pulling something alive out of that darkness. This is not science fiction. This happened. In January 2026, 3.2 billion years. Try to feel that scale. 3,200,000,000 years ago, Earth had no fish, no trees, not even a hint of the green, breathing planet we call home. There was only dark, scorching rock, and in its steaming shallow seas, something microscopic stirred. Something that needed no oxygen. Something that had learned to survive in conditions that would kill us within minutes. And then, in January 2026, a team of molecular biologists did something that until recently seemed like pure science fiction. They resurrected a molecular machine from that world. They didn't find its remains. They didn't build a computer model. They synthesized a living, functional enzyme, a protein molecule that last performed its work in Archean oceans, when a single day on Earth lasted only 18 hours. What is an enzyme? Imagine a vast factory where every station has a worker performing exactly one operation. That is how a cell works. And the enzyme is that worker. Without enzymes, life is physically impossible. They break down food in your stomach. They copy your DNA every time a cell divides. They participate in every breath you take and every thought passing through your mind right now. The resurrected enzyme was a special one, a nitrogenase, a molecule that fixes atmospheric nitrogen. Nitrogen is the foundation of amino acids and nucleotides, meaning the foundation of proteins and DNA. Without it, there is no life. But atmospheric nitrogen is an inert gas with a triple chemical bond, one of the strongest in nature. Breaking it is a colossal task. And yet ancient microorganisms learned to do exactly that, long before our planet gained an oxygen atmosphere. How do you restore a protein that hasn't existed for 3 billion years? The answer lies in molecular paleontology. The method is called ancestral sequence reconstruction. Scientists gather DNA from hundreds of modern organisms, identify similar genes, and literally run evolution in reverse like rewinding a film. Supercomputers reconstruct the original amino acid sequence of an ancestral protein. Then that sequence is synthesized, inserted into a living bacterium, and they wait. The enzyme worked. A molecule from the Archean seas moved again, resumed its function in a modern laboratory in the year 2026. Why? This is far more than idle curiosity. The first direction is Earth's ecology. Modern nitrogenases demand enormous amounts of energy, are sensitive to oxygen, and adapt poorly to extreme environments. The ancient enzyme is more robust. It was shaped in a world without oxygen, in chemical chaos. Understanding how it works opens a path to biological fertilizers produced without industrial synthesis. Today, nitrogen fertilizer production consumes roughly 2% of the world's total energy. Learning to do what ancient bacteria do could literally transform the planet. The second direction is the terraforming of Mars. Surface pressure there is less than 1% of Earth's. Night temperatures plunge to minus 125 degrees Celsius. But that is precisely what early Earth looked like. And that is exactly when organisms with those same Archean enzymes were alive. If we want to seed biological life on Mars, we need organisms capable of surviving in a frozen, nearly airless environment. Modern Earth bacteria are not built for that. But organisms equipped with reconstructed ancient enzymes, that is a different conversation entirely. These are potential pioneers. The first Martian colonists, not humans in spacesuits, but invisible microbes carrying molecular machines 3 billion years old. It sounds like the triumph of science, and it is. But this is precisely where a question surfaces that keeps some biologists awake at night. What if we are pulling more than just a protein out of the past? The Archean oceans were home to more than bacteria. There were viruses, an enormous diversity of viruses possibly richer than anything alive today. About 8% of the human genome consists of ancient retroviral insertions that wrote themselves into our DNA millions of years ago. The probability of accidentally pulling a fragment of an ancient virus alongside the target gene is vanishingly small, but it is not zero. And here lies the philosophical horror. Our modern biosphere is adapted to modern viruses. Our immune systems are the product of hundreds of millions of years of an arms race with pathogens. But a virus 3 billion years old has never encountered the immune system of a vertebrate. If something like that were ever to escape a laboratory, and that "if" is one scientists are obligated to take seriously, we would have no antibodies, no evolutionary memory, no prepared defense. This does not mean the experiments should stop. Science moves forward, and rightly so. But the level of biosafety must be flawless. Every step must be accompanied by international, transparent oversight. Questions that appear philosophical are, in practice, urgent and concrete. We are playing a role that only time has played before. For 3 billion years, evolution methodically filtered and transformed. Now we take its results and begin combining them at will. That is not wrong. It simply demands maturity. Power is not the same as wisdom. And yet, stepping back to see the full picture, it is breathtaking in its beauty. A molecule that remembers a world before oxygen survived geological eons, as encoded information passed from cell to cell for 3 billion years, and now it is at work again. Life on Earth is an unbroken thread, a thread stretching from the first chemical reactions in scorching hydrothermal vents of the Archean all the way to you, sitting in front of a screen right now. Every atom in your body has traveled through stellar explosions, through acid seas, through the bodies of creatures that lived and died long before multicellular life existed. You are the continuation of that molecular story. And for the first time, one link in that chain, us, has learned to read the past and carefully reach back to touch it. That is the true wonder of January 2026. Not the enzyme itself, but the fact that we could, that an intelligence grown from those same Archean oceans can now reconstruct their molecular language. If this video opened something new for you, or gave you a few minutes of genuine wonder at the magnitude of life, leave a like, and write in the comments what surprised you most. The resurrection of the enzyme, the idea of seeding Mars with ancient life, or the risk of ancient viruses? You can support the channel through Patreon. The link is in the description. Thank you for watching, for thinking, and for not being afraid to look into the depths of time. Stay with us, and until next time,